Hello friends, good afternoon and welcome to EduSite live lectures. Dear friends, today we are going to talk about processing of RNA. To discuss this topic, we have with us Dr. Varnendra Singh Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Hindu College, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome, sir.
Thank you very much. Dear friends, today I will be going to speak on a very important topic in molecular biology. This topic as per se deals with processing of RNA. RNA, as we all know, is a very important molecule which helps in transferring of information from DNA to the proteins. And as we all know, proteins are the important molecules which carry out all the functions inside the cell. The full form of RNA is ribonucleic acid. And as we know, it is a single stranded structure made up of ribonucleotides. But before it can transfer the information or it can carry out all the functions, this RNA has to be processed within the cell. And how this processing takes place is the topic which I am going to discuss today. As we all know that there are various types of RNA within the cell, but the three most important RNAs are tRNA, that is transfer RNA, mRNA, that is messenger RNA, and rRNA, that is ribosomal RNA. These three RNAs have different functions, but they work in tandem to carry out translation of mRNA into proteins. Now, tRNA, as the name suggests, is a transfer RNA, which is an adapter molecule which helps in bringing amino acids specific to mRNA codons to the translation site. The translation site is nothing else but ribosome, and the ribosome is made up of proteins as well as various rRNA molecules. These rRNA molecules are again transcribed inside the nucleus in eukaryotes, and in prokaryotes, since there is no nucleus, it is transcribed as such. The mRNA is the most significant molecules of all these, because whenever there is transcription of a gene, it is transcribed as a single stranded mRNA molecule. And this mRNA molecule is read in a triplet of nucleotides, which is also known as codon. For each specific codon, tRNA brings in a specific amino acid, and these amino acids are joined to one another, one after another, with peptide bonds, and this happens inside the within the ribosome. All these three types of RNA are transcribed, and before they can carry out their function, before they assume their structure, they undergo processing within the cell itself, within the nucleus itself in fact. So, what exactly is RNA processing? As I have already told, that RNA molecule which is copied from DNA template is known as a primary transcript. It is known as a primary transcript because after transcription, it will undergo various modifications, and only after these modifications and processing, it will become a mature RNA, whether it is a mature mRNA, mature tRNA or mature rRNA. So, this mature transcript is the one which is going to carry out the exact function of that particular RNA. Now, the RNA could be an exact copy of DNA, but in most of the cases it is not so, because whenever there is transcription, the DNA is transcribed directly. That means, whatever information is present in nucleotide, it is transcribed as a complementary nucleotide sequence. But after processing, there might be addition of new nucleotides, there might be removal of certain nucleotides, so that the mature mRNA which is formed after processing is not an exact copy of DNA. It has been processed and the sequence is differing from DNA. The processing of RNA involves various changes or various mechanisms, and these involve structural and chemical maturation of the newly synthesized primary transcript. These modifications may be essential for RNA to carry out its function, or they may also be required for regulation of gene expression. This modification which occur in RNA, they may occur during transcription, and during that time it is known as co-transcriptional modification, or these particular modifications may occur after transcription has occurred, and in this particular case it is known as post transcriptional modification. So, there are both ways in which modification can occur, that is during transcription or after the transcription has occurred completely and the primary transcript has formed. Now, there are various methods which lead to processing of RNA which are involved in RNA processing, and they can be summarized as under. The first method I will be talking about is cleavage. Cleavage is basically cutting off a long molecule, a long RNA into smaller RNA components, and this usually happens whenever there are polycistronic transcripts, that is a number of genes have been transcribed in tandem with one another, and this usually occurs in prokaryotes. In prokaryotes, various rRNA molecules are transcribed as a polycistronic molecule, and various tRNA are also transcribed along with rRNA as polycistronic transcript, and these transcripts are then cleaved to release individual rRNA molecules or individual tRNA molecules. And whenever rRNA has been transcribed along with tRNA, then cleavage leads to formation of individual rRNA and tRNA molecules. The second method which is involved in RNA processing is exonucleolytic degradation. Now, as the name suggests, exonucleolytic degradation is basically removal of nucleotides from one of the end of the premature RNA. This usually occurs when polycistronic RNA or polycistronic tRNA has been cleaved, and the individual molecules are then cleaved from one of the ends or both the ends to generate mature molecules, and this is known as exonucleolytic degradation. The third method is nucleotidal transfer. This is usually seen in tRNA maturation. The tRNA molecules which do not have a trinucleotide sequence CCA in them, inbuilt in them, they are provided CCA molecule through an enzymatic machinery. They are provided this trinucleotide to the 3' end, because it is essential for all tRNA molecules to have a CCA trinucleotide at 3' end. This is the end which will receive a specific amino acid for that particular tRNA. So, many tRNAs which do not have CCA, they will be provided with CCA by enzymatic transfer. The next method of processing is chemical modification of bases. Occasionally, there might be methylation in mRNA and rRNA, but it is a rare occurrence, and majorly the chemical modification of bases is seen in tRNA and snRNA. tRNA has a number of modified bases like pseudouridine and dihydrouridine, and snRNA also have modified uridine bases. Though mRNA bases can also get methylated, but the occurrence of such modification is less as compared to modification of chemical bases in tRNA. The next method of processing is nucleotide excision and replacement. In tRNA, it is seen that there can occur hypermodification of guanosine residues producing quercetin and viosine, and these bases are put inside the tRNA by excision and replacement method. The other methods of RNA processing are capping. Capping is basically seen in mRNA. Capping is a procedure of adding of a specific nucleotide to the 5' end of mRNA. In the case of eukaryotic mRNA, 7-methyl guanosine residue is added to the 5' end, and this helps in protecting the mRNA from degradation. It is important to point out that this particular processing or modification, that is addition of a 5' G cap, does not occur in prokaryote. The second modification which is prevalent in eukaryotic mRNA is polyadenylation. Polyadenylation is addition of number of A residues to 3' end of the eukaryotic mRNA. Again, this polyadenylation is not seen in prokaryotic mRNA. Another very important processing method for eukaryotic mRNA is removal of introns, and this particular processing is known as splicing. Splicing is seen only in eukaryotes, it does not occur in prokaryotes, because prokaryotes do not carry introns in them. Prokaryotes have only exons, and these exons are directly transcribed into mRNA, which is then translated into protein. Whereas in eukaryotes, exons are interrupted by introns, and these introns have to be removed from primary transcript to form mature mRNA, so that a functional protein can be produced. And the last and another important method is editing. Though this method of processing is not commonly seen in mRNAs, but it is of infrequent occurrence, and in this particular method there is change in the information carried by mRNA by base modification. Bases might get modified, or bases might get inserted, or bases might get deleted in the coding region of the pre-mRNA, that is the primary transcript. So, these are some of the methods which are basically carrying out the processing of RNA to make a mature RNA molecule. So, first of all I will be talking about processing of tRNA. tRNA is short form for transfer RNA. Some tRNA genes are transcribed singly, that means one gene is transcribed into a transcript, and that will be used as a carrier molecule for amino acids. Whereas many tRNA genes are also synthesized as transcribed as a polycistronic unit, that means a number of genes are transcribed in tandem with each other. This is usually seen in prokaryotes. In E. coli, some tRNA genes are co-transcribed with rRNA genes also, and it is a common occurrence again in prokaryotes, and this is not to be found in eukaryotes. In E. coli, the polycistronic tRNA or mixed tRNA and RNA transcripts are processed by ribonuclease enzymes. These ribonuclease enzymes act on the polycistronic molecule and bring about the cleavage of the individual molecule by making a cut at the 5' end. This particular figure depicts how tRNA molecules are transcribed as a polycistronic unit, and after that ribonuclease, which are also represented as RNAs, they carry out cut at the 5' end of these individual tRNA molecules. So, this is the figure which shows how RNase-P makes cut at the 5' end of the polycistronic tRNA transcript. The 3' end thus generated is now subjected to exonucleolytic degradation. That means, the individual molecule which are now generated, the 3' end of them are subjected to nucleolysis, and nucleotides are removed one after another starting from the 3' end, and this process continues till the CCA sequence is reached, because CCA is the acceptor sequence and it will be receiving amino acid for that particular tRNA. However, if the CCA is absent in that particular tRNA, that means the nucleotide sequence does not have CCA, then another enzyme known as nucleotidal transferase carries out addition of CCA to this particular tRNA. So, all in all, the effect of this particular processing is cutting off the number of tRNA molecules which are joined together into single units, and processing of these units into mature tRNA molecules which have functional ends, which have CCA ends, so as to take amino acid and become a mature tRNA. This figure shows how this is done. So, the first molecule is cut, that is RNase-P makes a cut at the 5' end, and after making the cut at the 5' end, another ribonuclease starts cutting this particular tRNA molecule from 3' end, that is RNase F makes a cut at 3' end. After the cut is made, one after another the nucleotides are removed from the 3' end till the CCA trinucleotide is reached. As we can see, CCA is present at the 3' end. So, this is a mature tRNA molecule which by the virtue of its nucleotide sequence has folded into a secondary structure, and this secondary structure is very essential for tRNA to carry out its function of bringing amino acids to the sites at ribosome during translation. In eukaryotes, the tRNA genes sometimes carry introns, and these introns have to be spliced from the transcript by a cleavage method, and these after the introns have been removed, there is modification of the ends and ligation. This particular splicing process do not follow the splicing process as seen in mRNA. Herein, a 5' OH group is generated after the endonuclease makes a cut, a 5' OH group and a 3' cyclic phosphate group is generated. After these particular ends are generated, they are acted upon by enzymes, modifying enzymes, which lead to formation of 5' phosphate group and a 3' OH group, which are later on joined by enzyme RNA ligase. It should be noted that the sequence of tRNA introns are not conserved. That means the splicing sequence is not conserved from one intron of one tRNA to another intron of another tRNA. The reliability of this method depends on the secondary structure formation of the tRNA, because the intron might form a complementary structure with the anticodon, which helps in cleaving the intron at the particular site. After this has been done, the pre-tRNA molecule is subjected to a number of base modification, and this process is known as post-transcriptional chemical modification, wherein bases like dehydrouridine, pseudouridine are formed after the pre-tRNA has been cleaved and a mature tRNA has generated. Also, hypermodified guanosine derivatives like eosine and viosine are inserted into the tRNA by nucleotide excision and replacement reaction. So, this figure shows how it occurs. Uridine bases can be methylated, leading to formation of ribosyl thymine, or there can be transformation of the linkage to ribosyl leading to formation of pseudouridine, and these bases make about 10% of the ribonucleotides of tRNA. So, it is a very high percentage of modified bases, and these modified bases are found in tRNA only after the primary transcript has undergone enzymatic processes. Also, the lower part of the figure shows how the intron, which is seen as yellow structure, is removed from this particular tRNA, leading to formation of a mature tRNA, and also the leader sequence, which is not required for proper formation of the secondary structure of tRNA, that is also removed from precursor tRNA, leading to formation of a mature tRNA with a CCA trinucleotide at 3' end. Now, after tRNA, I come to another important molecule, another important RNA, that is rRNA. rRNA is ribosomal RNA and is an important constituent of ribosome. It also shows catalytic activity for carrying out the translation of a given mRNA, and it is required for binding of mRNA to the ribosome. Now, both prokaryotes and eukaryotes synthesize polycistrionic rRNA transcript. Again, polycistrionic means rRNA genes are transcribed in tandem with one another. In E. coli, there are about 7 rRNA operons which contain genes for all the rRNA species which are found in this particular organism. It is also common for other prokaryotes, and this particular polycistron might also contain transcript of certain tRNA genes. The 16S ribosomal RNA and 23S ribosomal RNA, which are present in the polycistron, they form a secondary structure that is known as stem loop structure. This stem loop structure is essential for recognition by an enzyme known as ribonuclease 3. This ribonuclease 3 recognizes this secondary structure of ribosomal RNA, which is stem loop structure, and it cleaves that those particular ribosomal molecules, ribosomal RNA molecules, leading to their making them free from that particular polycystron, and individual precursors are formed. After these individual rRNA molecules are cleaved, then they are subjected to exonucleolytic processing from the ends, and after this processing has been done, it leads to formation of mature rRNA molecules. As can be seen in this particular figure, a polycistrionic RNA has been transcribed, and it contains transcripts of 16S, 23S, 5S and also of tRNA. Because of the composition of the ribonucleotides of this particular transcript, these particular rRNA form secondary structure, which are known as stem loop structure, as can be seen a ladder and a circle or a loop structure. This is known as stem loop structure. This stem loop structure is recognized by RNase 3, and it makes specific cuts in this particular molecule, generating individual rRNA molecules, which are further processed by exonucleolytic degradation from one of the ends, and it leads to formation of mature rRNA. In eukaryotes, the 5S rRNA is transcribed as a monocistronic unit by RNA polymerase 3, and it does not require any processing, because the complete gene has been transcribed and it is not transcribed in tandem with other genes. So, this particular transcript can directly be used for its function, it does not require any processing. Whereas, the RNA polymerase 1 synthesizes the 45S pre-rRNA molecule, which is required, which basically contains the 3 rRNA species, that is 5.8S, 18S and 28S rRNAs. This 45S rRNA, that is the primary transcript, it associates with a number of proteins which are known as ribonucleoprotein complex, and this complexing occurs in nucleolus itself. So, this particular complex is known as SNORP or processosome, and another name for this is small nucleolar ribonucleoprotein, and it can also be termed as SNO-RNP. So, this complex is basically a complex of 45S rRNA and protein. Some lower eukaryotic rRNAs also show self-splicing introns. That means, these introns will splice by themselves. They do not require assistance of any protein from outside. Just by their catalytic activity of the RNA, they carry out their own splicing. So, this figure shows how the pre-rRNA, that is the primary transcript of rRNA, is cleaved into specific molecules. Here 18S, 5.8S and 28S are cleaved from the primary transcript, and after exonucleolytic degradation, the mature rRNAs can be formed. This figure compares the rRNA processing of prokaryotes and eukaryotes. The upper panel shows the processing of rRNA of prokaryotes, leading to formation of 16S, 23S and 5S rRNA from a polycistrionic primary transcript, and the lower panel shows formation of 18S, 5.8S and 28S rRNA from a primary transcript of 45S rRNA. Now, I have already discussed the processing of 2 rRNA species in the cell, how it occurs in prokaryotes and eukaryotes. Now, I will be moving on to the third important RNA species, which is mRNA. Before I move on to that, I will just like to summarize the central dogma of the molecular biology. What does central dogma tells us? It is basically the principle of transcription of information from DNA into RNA and translation of information from RNA into protein. Sometimes this RNA can be reverse transcribed into DNA with the help of reverse transcriptase enzyme, but the basic flow of information remains in a single direction, that is the information which is present in the form of nucleotides, deoxyribonucleotides in DNA, it will be transcribed into the information of RNA, that is information in the form of ribonucleotide, and this information of mRNA will be read by the translation machinery, which is made up of ribosomes and tRNA, and according to the codons present in the mRNA, specific amino acid will be brought to that particular site, and one after another amino acids will be linked together by peptide bonds, leading to formation of a mature protein, which will carry out all the different types of function assigned to that particular protein. When we talk about processing of mRNA, there is a basic difference in the mRNA processing in prokaryotes and eukaryotes. In prokaryote, there is no nucleus, and whenever any gene is getting transcribed, as soon as the mRNA transcript, the mRNA transcript starts getting formed, it is also getting translated simultaneously, because there is no nucleus in prokaryote. So, there is simultaneous transcription as well as translation occurring in the prokaryote, and the mRNA of these prokaryotes is highly unstable and it is rarely modified. So, in that particular case, there is no processing of mRNA, whereas in eukaryotes there is primarily modification of the ends of the mRNA, first by addition of a cap at the 5' end, and after that addition of multiple A residues at the 3' end. After this has been done, the introns, which are a characteristic feature of genes of eukaryotes, they are spliced from the primary transcript, and the exons are spliced together, leading to formation of a mature transcript. Remember that there are no introns in the prokaryotic genes. Prokaryotes only have exons, so there is no need for splicing to take place or splicing to occur in the prokaryotic primary transcript. During the formation of mRNA in eukaryotes, it becomes associated with a number of proteins, and these are termed as heterogeneous ribonucleoproteins. The association between the mRNA and the protein is known as HNRNPs or heterogeneous ribonucleoprotein. Now, I will first be talking about the first change which any eukaryotic mRNA undergoes. This is known as capping. Basically, capping is addition of guanosine triphosphate residue at the 5' end of the primary transcript. This is catalyzed by an enzyme known as guanylyl transferase, and it creates a 5' phosphodiester bond between the newly added G residue and the first ribonucleotide of the mRNA. Usually, we know that the bonds between ribonucleotides in RNA or bonds between deoxyribonucleotides in DNA, they are linked by 5 to 3 linkage, whereas in this particular case the two nucleotides are linked by 5 to 5 linkage, 5 to 5 phosphodiester bond. After this particular guanine residue has been added to the 5' end, there is methylation of the seventh residue of the guanosine by an enzyme known as guanine methyl transferase. This is the second modification which the primary transcript undergoes. Usually in higher eukaryotes, it is seen that methylation occurs at the oxygen 2 of the ribose. That is, if the residue, that is the first residue is adenine, it can also get methylated at position the nitrogen 6 of the that particular adenine. So, after the addition of cap, there is methylation of guanine, then there is methylation at the oxygen atom of the first residue of the primary transcript. Also, if it is adenine, there will be methylation at the 6th position of that particular adenine residue. In some species, it is seen that the second residue of the original primary transcript, it is also methylated at the second oxygen of the ribose. So, these are the modifications which the primary transcript undergoes in conjunction with the addition of cap at the 5' end. This addition of cap is very essential because it helps in transport of the mRNA from the nucleus to the cytoplasm. As we know that the eukaryotes have a nucleus and the genes are transcribed into mRNA in the nucleus. This mRNA has to be transported outside the nucleus into the cytoplasm, and for that this particular cap is essential. Also, it is essential for binding of this particular mRNA to the ribosome. So, it is an essential feature which also prevents premature degradation of the mRNA. So, this figure shows how a cap is added to the 5' end of RNA. The right panel shows in blue the original primary transcript, and the left panel shows in brown the 7-methyl guanosine, which is added to the 5' end of the original transcript by 5' to 5' triphosphate bridge, which is not a normal bridge for any RNA, because the normal linking between two nucleotides is 5' to 3' link. The second modification which any mRNA of eukaryote undergoes is polyadenylation. Polyadenylation basically occurs at the 3' end of the mRNA, and this occurs after the primary transcript has been subjected to endonucleolytic cleavage, because whenever any gene is getting transcribed, it is transcribed much much more than the original length of the gene. That is, it is transcribed downstream of the original length of the gene. So, it has to be cleaved to bring it to the proper size. After that has been cleaved by endonucleolytic cleavage, there is addition of adenylate residues. The number of residues which are going to be added varies from one type of gene to another, and from 10 to 200 nucleotides of adenine can get added to a particular mRNA. This polyadenylation usually occurs at a specific site, and this occurs 10 to 30 nucleotide downstream, that is toward 3' of a highly conserved polyadenylation site, which is AAUAAA. This site is the signal that now polyadenylation should occur 10 to 30 nucleotide downstream from this particular region. A specific protein known as cleavage polyadenylation specificity factor is the one which recognizes the polyadenylation site, and an endonuclease carries out the cleavage reaction, and it is followed by action of polyadenylate polymerase, which catalyzes the addition of adenylate residue. The processivity of PAP is very low. PAP adds one nucleotide and falls off, then adds another enzyme adds another adenine residue and falls off. But after a few adenine ribonucleotides have been added, there comes another protein known as polyadenylate binding protein. This polyadenylate binding protein increases the processivity of PAP, so that a stretch of adenyl residues are now added by PAP. This polyadenylation is necessary because it may influence the stability of the RNA, and it may also play a role during translation. However, certain mRNAs like histone mRNAs are not polyadenylated. So, this is basically what occurs during mRNA processing. I have already talked about addition of cap, as can be seen in the second row. Cap has been added to the 5' untranslated region, and to the 3' untranslated region is addition of a poly A that is represented by A bracket N. After this has been done, any intron which is present in the this particular mRNA primary mRNA transcript, it has to be removed to make a sensible nucleotide sequence, which can then be translated into protein. If this intron is not removed, or if it is removed faultily, then there will be deleterious consequences for the organism, because even one nucleotide addition or excision from the exon sequence can lead to change in the reading sequence. If the reading sequence is changed, then the amino acid sequence of the protein is changed. If that is changed, the secondary structure and tertiary structure of the protein is changed. If that is changed, then the function of protein will be changed entirely. Might be that protein will become non-functional completely. So, this particular step is it has to have a very high fidelity, because if it is not highly fidel, then it can lead to very disastrous consequences for the organism. Now, the introns are the characteristics of eukaryotes, and as we know, prokaryotes do not have introns, but the number of introns in genes of different organisms also varies from the lower eukaryotes to higher eukaryotes. This particular figure represents the variation in the number of introns per gene in various eukaryotic species. On the left side are seen the lower eukaryotic species, and the rightmost side is represented by Homo sapiens, that is human. As can be clearly seen that the number of introns is very less per gene in lower eukaryotes as compared to higher eukaryotes. But there is no fixity of rule in this particular case also. The number of introns in genes within an organism can also vary from 1 to many. A single organism can be having a gene which is having 1 intron, and it can also be having a gene which can be having 30 introns. So, the number of introns and size of introns is variable between organisms and within organism also. Genes can be found which have exons as small as 150 to 200 nucleotides in length, and introns as long as 800 kb. And there are various such genes present in humans also. One of the genes of humans is dihydrofolate reductase. It is about 31 kilobase in length, and it has got about only 10% of the RNA which is in the form of exons. Only 2 kb is in the form of exons. There are 6 exons in that particular gene, and most of it is made up of non-coding RNA, that is introns. However, whatever portion of primary transcript matures into mature mRNA, that means all the nucleotide sequence which are present in mature mRNA will be termed as exons only, whether they are translated or not. So, the 5 prime cap region, the leader sequence which might not get translated, as well as the 3 prime poly A tail which is also not getting translated, are termed exons only. These are untranslated exons. So, these introns are removed by mechanism of splicing. The splicing can be done with the help of protein, and in that particular case it is known as spliceosome dependent splicing, or it can be an inherent feature of the RNA itself, that is known as self splicing. And there are various cases of self splicing, but most of the mRNAs are undergoing splicing with the help of spliceosome. Now, this splicing has to be very accurate, because addition of a single nucleotide to the exons or removal of even a single nucleotide from the exon can change the entire reading frame, as I have already told you. It can be deciphered from this particular example, though it is for the layman, but it can be done from here. As we can see that there are letters in black and letters in brown. The letters in brown, when they are put together, they will make some sense. Like we see there is dog and letters in black which do not make any sense, ate the bun. So, there is one intron that is black letters, and there are 2 exons which are brown in color. Now, if we take out the black letters properly, if the black letters are excised and the brown letters are joined together, they will make a sentence: dog ate the bun. So, exons are also read as trinucleotide sequence in the form of codon. Against each of the 3 letters which are properly read, there will be brought an amino acid. If any single mistake is made, like suppose if the cut is made so that one black letter comes along with the brown letters, so the entire sequence change. Now, it becomes dog, tat, eth, ebu, and n. So, it is not making any sense. Similarly, if one of the letters is excised from the exon region, that is the brown region, again the after splicing, the sequence of the letters changes and it does not make any sense. In the case of mature mRNA, the triplets might make a sense after splicing also, but that will lead to change in the amino acid which are brought into that particular protein, and this will lead to change in the structure and function of the protein, or this might lead to formation of a stop codon. And if stop codon occurs in the mature mRNA, then the translation process will stop abruptly. There will be abortion of translation, and that particular protein will not be formed. So, this particular process has to be carried out with high fidelity. And how this is done? This is done with the help of certain conserved sequences which are present between intron-exon boundary. Now, conserved sequences are those sequences of nucleotides which are invariant between species and within species in all the genes. So, all the eukaryotic species, they have conserved intron-exon boundaries, and in this particular figure we can see that in the exon and intron boundary we have GU, and the intron and exon boundary we have AG. So, GU region and AG nucleotide region, these are conserved site, and these are basically known as 5' splice site and 3' splice site. Another important conserved region is that of the branch point site. This branch point site is more towards the 3' region of the intron, and it contains an adenine residue which will be responsible for carrying out the splicing reaction. This adenine residue is followed by a number of pyrimidines. So, all in all, the conserved sequences are the ones which are required for carrying out splicing of introns from the exons in a primary transcript of mRNA. So, how this particular splicing reaction occurs is seen here. There are two trans-esterification reaction occurring whenever splicing has to occur. Basically, the first reaction is between the adenine of the branch point site and the residue at the intronic boundary. As can be seen here, the 2' OH group makes an attack, nucleophilic attack at the linkage between the two nucleotides at the exonic and intronic boundary. This 2' OH attack leads to excision of the first exon from this particular structure, and after it has been excised, 3' OH group becomes free. This 3' OH group makes an attack again at the intronic exonic boundary nucleotide, the linkage between the two nucleotides, and the two exons which are represented by black solid lines, they are spliced together, whereas the intron comes out in the form of lariat. Lariat is basically a noose or a lasso like structure, and this particular intron is then degraded. So, this particular reaction does not require any energy consumption. There is no consumption of ATP. However, the proteins which are required for bringing about this particular reaction, they require a lot of ATP for their assembly. And secondly, it is to be noted that this reaction is reversible also. That intron lariat can again go back and get inserted between the exons. But that rarely happens in a cell because first of all, there is increase in entropy. One RNA molecule is getting cleaved into intron and spliced two exons. That means one is getting cleaved into two. Secondly, one of the product, that is intron, it gets cleaved immediately after formation. So, there is no reversible reaction occurring here. The intron does not go and insert itself into spliced exon anymore. So, basically this is the formation of the spliced exon by two trans-esterification reaction. There is no energy consumption because two bonds are getting broken and two bonds are getting made. However, this complete thing requires assistance of a complex of proteins which are known as spliceosomes. So, this figure basically represents how an attack is made by 2' OH to the 5' end, and after that how a 5' OH groups make an attack at the 3' end of the intron-exon boundary. So, we saw that the cis splicing, that means the exons within a primary transcript are getting spliced together, but trans-splicing can also occur where two different RNAs can get spliced together by the similar mechanism. However, in this case the exons of two different RNA will get spliced together, and the intron which will be formed will not be a lariat shaped structure, it will be a Y shaped structure, and this is frequently seen in the mRNA of trypanosomes and other lower eukaryotic organisms. The splicing was discovered in as late as 1977. Earlier it was believed that genes are transcribed and the RNA is translated into protein without any processing, because the process was not known. However, some scientists worked on adenovirus, which is a virus of humans, and they wanted to find out what are the sequence of different mRNAs which code for different proteins of this particular virus. when they isolated different mRNAs, and they found that each of them were having similar 5' sequence. So, they came to a conclusion that the complete genes of the this particular virus are transcribed as a single unit with a single 5' sequence, and after that there is excision of specific exons, so that for a particular protein, a particular exonic sequence mRNA is generated. And this they they came to conclusion using a certain experiment wherein they took DNA of the virus and they hybridized it with the RNA of that particular virus, different RNAs of virus. As can be seen in the lower panel, whenever there was a denaturation of the double stranded DNA, the single stranded DNA made complementary base pairing with the RNA, and it can be seen that the RNA bound to specific regions made complementary base pairing with specific regions of DNA, whereas those regions which were not complementary to the RNA, they were looped out from that particular region. And here we can see that various introns are looped out, so that it signifies that the RNA of this particular virus, it has been spliced together, the exons have been spliced together and introns have been removed from this virus. So, this was the discovery which led to finding out that there is processing of mRNA in eukaryotes. So, this is the figure, this is the original experiment, the electron micrograph of the original experiment carried out by Burgett et al. In 1977, which established that RNA is binding to specific regions of DNA. It is not binding contiguous regions, it is binding to only those regions with which it shows complementarity. Now, coming to the machinery which brings about splicing. This machinery is known as spliceosome machinery, and it is made up of a number of proteins and snRNA. snRNA is basically small nuclear RNAs, and in totality it is made up of about 150 proteins and 5 RNAs. These 5 RNAs are termed U1, U2, U4, U5 and U6, and collective term for them is small nuclear RNA. They are termed as U because the uridine residues in these small RNAs are highly modified. Each of these RNA in these spliceosome is between 100 to 300 nucleotides long, and it is found complexed with various proteins, and together they are known as snRNPs or small nuclear ribonuclear proteins. However, they are pronounced as SNRPs. Now, this figure shows secondary structure of various spliceosomal snRNAs. U1, U2, U4, U5 snRNAs are transcribed by RNA polymerase II, and they have a trimethyl cap G cap present at the 5' end, and they are bound by SM protein. Whereas the U6 snRNA, it is transcribed by RNA pol III, and it is having unusual cap, and it is not bound by SM protein. Because of the specific sequence of these snRNA, they have a specific secondary structure, and this secondary structure is essential for carrying out specific binding to mRNA and the catalytic process. Also, it is responsible for binding of other proteins to spliceosomal complex. Now, the exact makeup of spliceosome differs at different stages of splicing. It is not that all the snRNPs, that is U1, U2, U4, U5, U6 are at a single time bound to mRNA and carry out splicing at a single go. There are various steps for this particular splicing to be carried out, and it requires help of various other proteins which are loosely associated with spliceosomal complex. The snRNPs carry out important function of recognizing the 5' site, recognizing the branch site, bringing the 5' site and the 3' site together, carrying out the first trans esterification reaction, and then carrying out the second trans esterification reaction. It helps in joining the exons together, and this it does with the help of specific characters of the RNAs of the snRNPs. As can be seen here that the snRNA of U1 snRNP is complementary to the 5' splice site of the mRNA. This site of mRNA is also complementary to snRNA of U6 snRNP. Secondly, U2 snRNP has snRNA which is complementary to the branch point site of the mRNA, which leads to exuding out of the A residues. After which it carries out trans-esterification reaction. The C figure shows that U6 RNA is complementary to the U2 RNA, and when these two RNA make complementary base pairing, they create active site which leads to the first trans-esterification reaction. And the D figure shows that the branch point site is the site where the first branch point binding protein BBP can bind, and later on it is again recognized by U2 snRNP after BBP has moved out from this particular site. So it is seen that the sequence of snRNA is very essential for recognizing specific sites in the primary transcript, so that splicing can be carried out effectively and without any fault. There are three major types of spliceosome: major spliceosome, minor spliceosome, and trans-splicosome. So major spliceosome I'll be talking about first. The major spliceosome is the one which carries out the splicing of the most of the mRNA in eukaryotes, and it requires presence of U1, U2, U4, U5 and U6 snRNA. So the first interaction is between the U1 snRNA, which binds to the 5' splice site with the help of U1 snRNA. After this is bound, U2AF, that is U2 auxiliary factor, binds near the 3' splice site, and it has got two subunits. These two subunits help in binding of another protein known as branch point binding protein to the branch point site. After this has been formed, this particular complex has been formed, the branch point binding protein moves out from the branch point site, and the U2 snRNA comes and binds to the branch point site by the virtue of its complementarity with the mRNA. This complete complex of U1 and U2 and along with U2AF is known as pre-spliceosomal A complex. After this complex has been formed, the U2AF auxiliary factor moves out from its particular location. After U2AF factor moves out, there will be input of tri-snRNP, that is U4, U5, U6 snRNP to a primary complex. The interaction of U1 with 5' splice site and U2 with branch point site is basically dependent on the sequence of the mRNA. If any mutation is carried out in either of the snRNA or pre-mRNA, so that there is no complementary base pairing, then proper excision cannot be carried out. So after U1 and U2 have come to their particular location, U6, U4 and U5, that is tri-snRNP, come to that particular site, and after this happens, there will be movement of U1 out from the 5' splice site. U6 is bound to U4 by specific complementary reaction between the RNA of these two snRNPs, and after U6 is bound to the site, the 5' splice site, the U4 also moves out. So we have now U1 and U4 moving out, and we have only U2, U5 and U6 present along with mRNA. Now the U2 and U6 snRNPs are brought together, and they bring about the catalytic reaction, that first trans-esterification reaction. The first trans-esterification reaction leads to cleaving of the first exon, and after that U5 helps in attacking the second exon, and there is splicing between two exons, and the lariat shaped intron is moved out. After the lariat shaped intron has moved out, it is linearized and then it is degraded by the nucleases present in the cell. So basically what happens is this is the splicing pathway. This is the splicing pathway which shows that U1 and U2AF and BBP, that is branch point binding protein, are the first one which complex with the mRNA. After that, branch point binding protein moves out, and it leads to complexing of the U2 at this branch point binding site. When U2 binds to the branch point binding site, then A, that is adenine residue, is exuded out, and after it exudes out from this particular location, it can carry out first trans-esterification reaction. But before that, there is movement of U2AF factor out from the 3' branching site. After U2AF is moved out from this particular site, there is attachment of tri-snRNP particle, which is a complex made up of U5, U4 and U6. U4 and U6 are interacting with each other through complementary base pairing of their RNA, whereas U5 is loosely bound to them by protein-protein interaction. Once this complex comes into place, U1 moves out from the 5' splicing site, and in its place comes U6. As I showed you that there is specific sequence of mRNA which binds both U1 and U6. So, at one time only U1 or U6 can be bound to that particular location. So, U6 comes and binds here. After U6 comes and binds, the interaction between U6 and U4 lessens. So, U4 also comes out from that particular location. So, now we have U6, U5 and U2. By interaction between U6 and U2 RNA, a catalytic site is created. When this site is created, the first trans-esterification reaction between the adenine at the branch point site and the intron-exon boundary is done, which leads to formation of the splicing of the first exon. And once the exon is excised, the U5 snRNP helps the 3' OH group of the first exon to make an attack at the intronic exonic boundary at the 3' end. And once this is done, the two exons are spliced and the intron in the lariat form is taken out, which is subsequently degraded. So, I will stop with this at this particular point, and in the next lecture I will be talking about how splicing affects various phenomena which occur in the cell. Thank you.
I would like to thank sir for this very informative lecture, and I hope friends that you find this lecture very benefiting and very informative. On that note, I would like to thank you people as well for watching. Stay tuned and keep watching. Thank you.